a tick. Talk to the tune of a hickory stick. You were my queen in calico. I was your bashful barefoot beau, and you wrote on my slate, I love you, Joe, when we were a couple of kids.
Hi, I'm back again, and you're back again. That's because you enjoy this class. The purpose of this activity is to identify the similarities and differences between plant cells and human cells. A cell is the smallest unit of life. A group of cells makes tissue. Today we're going to look at onion cell. You need to take out your materials for this experiment, and it includes your slides, the cover slip, Lugol's solution, which is going to dye the nucleus of the cell, and you need an onion. As you can see, this one was cut in half, and there is a membrane. The membrane's right there, and you just take your forceps and pull out the membrane, but you need to flatten it. You need to make sure it's flat and not folded over. Right there in the middle is a flattened portion. I'm going to add a drop of Lugol's to dye the nucleus, which is the brain of the cell. I'm going to put a cover slip over it and press down on the cover slip, and you can move that onion membrane. Remember, the membrane is not one cell. It contains many cells, so that means that it's multicellular. Many cells. Now you're ready to put it under the microscope. Turn on the light, looking through 4x. Look for the brownish color on your cell and focus in with 4x, and you will see a beautiful picture of the onion membrane with all its nuclei. Looking at the onion cell, you will observe the following. A cell which is the smallest unit of life. Looking closer, you will see a group of cells making up the onion cell membrane. The onion membrane is composed of many cells. This makes the onion membrane multicellular. A multicellular membrane contains many cells attached to each other. As seen here, you will see the cell wall attaching to each and every one of those cells. Therefore, the onion having many cells makes it a tissue here. The membrane is composed of many cells, and a tissue is made up of many cells. As we continue viewing this picture, notice the cell walls, which are the outside border of each cell. The cell wall determines the shape of the cell. The potassium iodide or Lugol solution has stained it brown. There is also a plasma membrane lying adjacent to the cell walls. You are not going to be able to see the plasma membrane. The cell wall, though, protects the plasma membrane from bursting because of the intake of water. Notice the nucleus. The nucleus is found in each cell. It is brown in color and circular. Each one of these nuclei, plural for nucleus, control the cell actions. Without a nucleus, the cell would die. There are many structures or organelles that cannot be seen with your microscope. This includes the plasma membrane, as mentioned before. The chloroplasts are not seen either. They are unique to plant cells. Their special function is to provide photosynthesis. What is photosynthesis? Photosynthesis is a process whereby food is made. The food is glucose, also known to be a sugar. The large vacuole in the center of the cell pushes the brown nucleus to the side or to the border of the cell. The nuclear membrane is a cell membrane that encloses the cell nucleus. It is not seen here, but it has holes or pores located on the membrane to permit the passage of certain materials like proteins between the nucleus and cytoplasm. The vacuole and remaining part of the cell outside the nucleus contains cytoplasm. What is cytoplasm? This is a thick solution that fills each cell and mainly composed of water, salt, and proteins. I will now compare a plant cell to a human cell. Plant cells have a cell wall. Animal cells do not. It only has a plasma membrane. You will see this in the next laboratory session. The plant cell has a large vacuole, while the animal cell has a small vacuole. Both have nuclei, but the human cell has it in the middle of the cell because of the small vacuole. Finally, the plant has chloroplasts, while animal cells do not. They cannot make their own food. Okay, we are all ready to record all our observations in our laboratory notebook. This is laboratory number 3, session 3. There's an introduction, followed by materials, followed by instructions on how to make the slide. The slide is a wet mount. Include the forceps in materials, the onion, the cover slip, results and conclusion. Now you are to draw what you see under your microscope and compare it to my drawing. Now you are to label the organelles you see. Nucleus and the cell wall. What we didn't see was a large vacuole, the nuclear membrane, the plasma membrane, the chloroplast, and cytoplasm. The function of the organelles that are not seen should be represented by a dotted line. The organelles need to be listed with their function, with their respective function. And let's go back to the microscope. Okay, magnify the initial magnification on 4x. And then as soon as use the coarse adjustment to do the major focusing. And then switch to 10x or 10 times 10, which is 100x total magnification, and do your fine adjustment. And you just have to turn it very little. And there you have your drawing that you will make. Take out your biology notebook. Label the lesson on onion cells. Draw the onion cells as seen under your microscope. I'm looking at what all of us should see under our microscope. There are many cells here. Remember, many cells means multicellular. And there are many nuclei here, more than one nucleus in the picture. So let's go over where the nuclei are. See it on the outer side here? And then see it on the border there? Here it's on the border. Here it's on the border. Here it's on the border, pushed to the side. All of them are pushed to the side, as you can see here. All of them are pushed to the side and are not centered. And this is because, again, of the vacuole, which is large and pushes it to the side. When we go over the human cell, you will see differently. You will see the nuclei in the center because there's only a small vacuole. You do not have to copy my diagram. Use your imagination and draw out the organelles which are seen and not seen. There's a dotted system to show you the ones that cannot be seen. The ones that cannot be seen, you will see, are the chloroplasts, the large vacuole, the nuclear membrane, and the cytoplasm. Notice the cytoplasm is shaded in and marked with a dotted system. The organelles that you can see under your microscope consist of two. One is the cell wall, and the other one is the nucleus. Happy drawing! And we're going to shut down our microscopes and clean up. Put the ocular lens back to 4x. Take off the slide. Turn off the light. Get some lens paper and clean off the ocular lens that you use. So I just clean them all just to make sure that they didn't get any water or dye on them. And you got probably dust on the ocular lens. Clean that up as well. And now we're ready to put away the microscope. Put the cover over the microscope. Unplug the cord, or the cord may be attached to the microscope. In this case, my plug is not attached to the microscope. I have to wrap it by itself. Now, if it's attached to the microscope in your case, what you do is wrap it around the base of the microscope. So, hasta luego.